Good evening, listeners. Welcome to the April 2022 episode of Archaeology in Connecticut. I'm your co-host, Scott Brady. I'm here in the studio. Don't worry, Sarah's on the line, and we'll get to her very shortly. But I'd like to take a quick moment to introduce the new president of the Friends of the Office of State Archaeology, and Glenda Rose. Welcome to the show, and congratulations on the presidency.
Thank you, Scott. It's pretty exciting to be the new president of FOSA. It's a really great group. I worked with a lot of people. I've enjoyed it. I joined in 2012 with Scott when we both retired. So it's been fun. I've facilitated a workshop or two and done a few other things. So I'm looking forward to digging in. Just as a quick background, my childhood interest in archaeology was sparked by my dad. I grew up in Westford near the Westford Bottle Factory, and we had a bottle dump on our property. And we dug there and found a lot of beer bottles, and no flask, darn it, but the beer bottles. My mother found a pretty quartz point in the garden and a couple kaolin pipes, and my dad had a great point collection from South Windsor and Manchester. He had this really neat paint pot. Don't know where he found it, but it was a really nice collection. So it really got me interested in archaeology and history, and I really love it. And now a lot of other things merge with the archaeology group. I belong to the Mishamasek Hiking Club. I'm the historian and newsletter editor. And we see a lot of features on the trails that, again, I get really interested in. So I love archaeology, and I'm looking forward to working with the group, supporting Sarah, and supporting the membership. And any input is welcome. Thanks.
Well, yeah, thanks. And, I look forward to seeing what you do with the organization. I know how passionate you are, and it's going to be really exciting. Now, I want to reach out to Sarah. Sarah, this show is going to be really interesting. You and I have talked about this for quite a while, and you know how excited I was to hear about this. Do you want to take it and introduce our guest for tonight?
Sure. Hi, everybody. Thanks for joining us for the April episode of the Archaeology of Connecticut. Tonight, we're joined by our special guests, Dr. Deborah Bolnick, Dr. Raquel Fleskes, and Samantha Archer from the UConn Anthropology Department. And we're going to have a discussion about DNA research and archaeology. We'll talk about the basics of DNA and DNA analyses and talk about the different intersections of DNA work and archaeology, looking at genetic research and work that is geared more towards kind of social justice aspects, including identification of burials and working with descendant communities. Before we get started talking about our topic for tonight, though, I'd like to take a minute and meet our guests. So, Deborah, Raquel, and Sam, would you mind just telling us a little bit about yourselves and your work?
Yeah, to start, I'm Deborah Bolnick. I am an associate professor of anthropology at the University of Connecticut and also affiliated with the Institute for Systems Genomics. And I am trained both as an anthropologist and as a geneticist, and I work mostly in indigenous communities, looking at DNA from both living individuals and ancestors to learn how the DNA, or to look at how the DNA can help us reconstruct human histories, community histories, and understand more about the relationships and the ways that community genetic makeups have changed over time in association with social factors and historical events.
Great. Hi, everybody. My name is Raquel Fleskes. I am Dr. Deborah Bolnick's postdoc. My research falls in the intersection of using ancient DNA or DNA extracted from archaeological burial sites to think deeply about ancestry and relatedness and how this information can be used to deepen site interpretation. So my work largely intersects with Deborah, but also works more specifically with European descendant communities in the colonial Chesapeake region. So we're doing work with Jamestown and the 1607 Burial Ground Project, which I'd be happy to talk a little bit more about later on, as well as with African descendant communities, such as in Williamsburg, Virginia. We're working with the First African Baptist Church Project to do some community-based work that centers around trying to identify and really bring information on lived experiences of the individuals that are interred at that site. And I've also done a lot of community work down in Charleston, South Carolina, which is a little bit farther south from our Connecticut focus, but helps us really think about how we do this type of work that is reflective of the histories of genomic research, meaning in the histories of medical research on African American bodies, and how we go about this work that's really cognizant and integrative of descendant communities' perspective. So I'm really happy to be here and really excited to take part in this conversation.
Hi, everyone. My name is Sam Archer. I'm a fourth-year doctoral student at UConn in the Anthropology department. My master's degree is from the University of Texas at Austin, and that project, my master's thesis, focused on the kind of a critical meditation on how we determine sex and gender from ancient genomic data in an indigenous population in what is now Belize. And then my dissertation project is focusing on two mid-19th century cemetery populations in Texas, one in the city of Austin and one right outside of the city of Houston. And I, like Raquel, focus on individuals with European and African ancestry, and I'm interested in trying to understand how the state of Texas's formation as kind of an entity can be understood through the genomic diversity that we see in the individuals that I'm looking at for my dissertation.
Great, thanks so much. So I really want to talk more about all of your research in more depth in a little bit, but I think that maybe to start, we should maybe cover some of the basics about DNA research. So I guess one question that I have is that maybe if you could explain to our audience kind of the relationship between ancient DNA research and archaeology and how that all fits together.
Yeah, I think this one is going to me. So this is Sam. Okay, so the relationship between archaeology and ancient DNA is still an in-process relationship. Ancient DNA itself is still a relatively new field, especially in comparison to archaeology. So if we understand archaeology as a field that's been developing at least over 120 years, possibly longer, depending on how you understand the development of the field, ancient DNA has only really been around as a field of study for about 20 years, right? So we're looking at a relatively new science versus a science that is very well established. And it's only really been in the past couple of years that the technology has developed to the point that sequencing ancient genomes is becoming more and more cost-effective, and it can be done a lot more quickly than it could 20 years ago. So today in 2022, we do the technology to sequence genomes from archaeological materials much faster than we could 20 years ago. And because of this rapid increase of production and capability, and also because there are more and more laboratories that have been built in the past decade with the capability of working with ancient DNA, we're kind of in the midst of a kind of scholarly revolution in terms of collaboration between archaeology and ancient DNA. And I think both fields are realizing that there's a need for the two fields to pay more attention to each other, especially because it's in my experience that archaeologists tend to see ancient DNA as inaccessible to them, and ancient DNA researchers still tend to be a little too attached to their laboratories. But the two fields are starting to look towards each other. And I specifically think this is really important that we do this more collaborative and integrative research especially because we make the best scholarship with genomic data from archaeological materials with archaeological context fully incorporated from the beginning of the research design to the publication. So again, it's a growing and constantly developing relationship between the two fields, but we are seeing very recently more interest in collaboration in both fields.
Can I jump in for a second? And, our audience is pretty diverse, and, we have professional archaeologists, and we have, the average person that is just interested in archaeology. I think we're all familiar with, the DNA tests that we can all do now through mostly genealogical websites. What's the same or what's different about that, and what we're looking at, what you guys are looking at in this ancient DNA research?
Yeah, I can take that one. This is Deborah. I think there are some similarities. In both the kinds of direct-to-consumer genetic tests that you can buy from a company like 23andMe or Ancestry.com. These are tests that are looking at an individual's DNA. This is the hereditary material, that kind of double helix string of A's, T's, C's, and G's that are found inside each of the cells in our body. And I think the common ground between the genetic tests that we can buy as consumers and DNA that we're studying from ancestral individuals is that in both cases we're looking at the sequence of letters in particular regions of the DNA and comparing those sequences, that string of A's, T's, C's, and G's, from one person to another. What we see is that most of that sequence is actually identical in all people, but there are some spots where those letters, those molecules differ from one person to another. I might have an A at one position in my DNA, Sam might have a T at that position. And these points where there are differences are often referred to as genetic markers because they're marking places in the DNA that are different from one person to another. And when we're looking at DNA with these DNA tests today, we're using them to look for similarities and differences among individuals. Related individuals usually inherit the same genetic markers from a shared ancestor. And so the tests that are available to us through these companies today can be used to identify closely related individuals or closely related groups of people because individuals will exhibit the same genetic markers in their DNA. Similarly, when we're looking at ancient DNA, what we're doing is extracting DNA from the remains of individuals who lived in the past. And this could come from a person's hair, from their bones or teeth, from dental calculus or kind of calcified dental plaque, surfaces of an individual's teeth, and some other sources. And by looking at the sequence of letters in that individual's DNA and comparing that sequence to other individuals who lived in the past or individuals who are alive today, we can make some assessments about relatedness, about whether two individuals who lived in the past were closely related to one another or whether one of those ancestors was closely related to someone alive today, just as you make those comparisons through these tests that are available commercially. And so they can be really useful, provide some useful insights into relatedness between individuals. And then if you learn that a person is related to individuals who live in another part of the world, that can then shed some light on the ancestry of that individual as well.
Thank you. That makes a lot of sense. So it's really fairly similar, although you're looking at the time period that's really the difference, right?
Yeah. With ancient DNA, you have this ability to look not just at DNA from people who are alive today, but at individuals who are living in the recent past and also in the much more distant past, because we can look at DNA from individuals who lived a few decades ago, several hundred years ago, and even in some cases tens of thousands of years ago.
So can we talk a little bit about the process of DNA analysis? Maybe talk about how DNA is extracted from human remains, how you actually get the material, and what are the challenges of that when dealing with archaeological people and populations that may sometimes come from, backgrounds that have kind of a challenging relationship with anthropology and genetics and all of that. But, if we could just talk about the methods a little bit, how the DNA is extracted from the human remains to start with, and then maybe move on to some of the other aspects of that.
Yeah, sure. So this is Sam. I'll just talk a little bit about how ancient DNA is actually extracted from human remains. And then I think Deborah and Raquel can jump in regards to the other questions. So a lot of folks, when they first one of the very first questions we get about extracting ancient DNA from human bone material is this process destructive? And I think that there is a general understanding that in order for us to get DNA from human bone material, that means the bone will be completely pulverized or destroyed, and that's not necessarily the case. It really depends on the particular laboratory's protocols, but it is actually possible to do very minimally invasive extraction procedures that some folks consider to be completely non-destructive, and some folks would call it just very minimal destruction. So, for example, one of the best sources for human DNA, human ancient DNA, is teeth. And so in our lab at UConn, we use a protocol that allows the tooth to, it just soaks and rocks in what we call a digestive buffer overnight. And so some heat is applied, and through the chemicals that are in this digestion buffer, this actually allows the DNA to be pulled out of the tooth root, and there's no overt damage done to the actual bone. So this might result in the tooth looking a little different once it's dried, but in my experience, you generally can't tell that this procedure has been done to it. So that is one of the best ways to do a minimal destructive extraction process. And then if teeth aren't available, it's also possible for us to take a small drill to the bone material and shave off just a really small amount of bone powder that is like roughly equivalent to what you would find in a pill. So we're talking about a very small amount of bone powder. And obviously this does incur direct destruction to the bone material, but it is still minimal. And it's usually possible to drill as inconspicuously as possible. So we tend to try to do it in a place where it's not as noticeable. And so obviously that is a directly destructive method, but it doesn't require quite as much destruction as some folks think it means, like, completely powdering the entire bone to a pulp. And so researchers are constantly trying to develop newer methods to get ancient DNA in the least destructive ways as possible.
And I'm going to piggyback off of what Sam has talked about with the minimally destructive, and we know that sometimes, though, in archaeological context, human bone may not be very well preserved. So we have things like if you have really acidic soils or high water content, it can really damage the preservation of the bone. And so in cases where, let's say, there's not really good preservation of teeth, researchers have also begun to target a part of the bone, part of the skeleton called the petrous bone, or we could also hear this called the petrous temporal or the petrous pyramid. And this part of the bone actually encases our inner ear bones, right, our auditory canals. And it contains really, really, really dense bone. And so in ancient DNA, usually it's the harder the bone, the better the DNA preservation, which is why things like tooth roots are also really great for ancient DNA, because our teeth roots are very, very dense parts of our skeleton. So in cases where researchers do decide to do more destructive means of testing, like what Sam was talking about with the drilling, where we do powder the bone, the petrous is a really great candidate for getting really high concentrations of DNA, right? We call this endogenous DNA, or DNA that's belonging to that archaeological individual. Now, what's tricky is that this petrous bone, because it's in our inner ear bone, it can be really hard to access. So we can either access it through the bottom of the cranium or the skull. You can drill into that way, or if the skull is fragmented already, we can access it through the inside of the cranium. So this is a new method that has kind of come out the last maybe 4 or 5 years. So it results in, we do the same types of processes that Sam talks about, where instead of soaking the tooth itself, we would just take bits of this bone powder and then soak it in that extraction buffer to pull the DNA out. And then we use a lot of different other types of solutions to basically remove all the other stuff that gets pulled out with that DNA. So not only DNA are we extracting, we're extracting proteins and salts and maybe fungus and all this, whatever is present in the bone. So we also have to basically select only for that DNA, and we use something called a spin column. And it's basically a very little tiny capsule that goes in, where it goes into the tube, and the solution will pass through this capsule, and then the DNA gets stuck in a membrane. And we are able to basically clean it, remove all the salts, remove all the proteins. And when we're ready and that DNA is cleaned, we'll put another solution into it to release that DNA from the membrane. So we've isolated everything from that bone sample and are only left with DNA. And with that part, then we can move forward and move to preparing the DNAs for sequencing.
You talked about
go ahead, Scott.
Yeah, you talked briefly about the tartar and calculus on teeth. Are there other kinds of non-bone related, besides hair and bone, materials that you can get the DNA from?
Oh yes, I'm so glad you brought that up. Dental calculus is a really interesting type of DNA extraction. It uses the same methods that we're talking about right now, which is we have to put the bone in a solution for overnight or 24 hours to leach out or pull out that DNA, but we can change the type of substance that goes into that solution. So, for instance, dental calculus is basically these layers of fossilized plaque, and plaque in itself, that biofilm, is an extremely hard substance that traps all these different types of our microbiome that's in our oral cavities, in our mouth. So what we can do is basically take a little bit of that dental calculus, and I'm talking like a very, very small amount, like, like a little piece of a fingernail almost, and you put into that solution. You basically would just grind it up into a small powder, and then you would follow a little bit of a longer incubation time, so it would be 5 or 6 days instead of 1 day, because the calculus itself is much tougher. So it takes a little bit longer to penetrate in and pull out that DNA substance. Now, for things like, let's say, soil DNA or hair, let's say this is a mummified individual, you would follow a slightly similar protocol, but again, it would be modified. So with soil extraction, this is a whole brand new field that's just rapidly emerging, where folks will take soil samples from around, let's say, an archaeological burial, and they want to basically characterize what the little bacterial communities around that burial are. And you can take your DNA, your dirt, your pieces of dirt, and you would basically soak it for a while and then pass it through a bunch of filters. Now, because dirt, obviously, is very dirty, we have to make sure that we're only isolating those microbial DNAs and not, let's say, like of salts or other things that can be found in the soil. So I think this is a really rapidly growing area of the field where DNA can not only come from human skeletal bones, but all of the organic substances that are part of the body and that we interact with during the processes of death and decay.
So once you have the DNA extracted, what kinds of information can a person's DNA provide about their ancestry? And I know that Sam earlier mentioned their lived experiences. What can you actually get from DNA analysis? Like, what can it do and what can it not do? Because I think people tend to think of it as sort of a, almost like a magic kind of thing that provides all this information. But what are the limitations and what is possible, I guess, with DNA research?
I can take this one. So there's a lot that we can learn from the DNA, but we're also limited to some extent. I guess maybe to start, the DNA can tell us about some general broad biological characteristics of a person in the sense that it can help give us some insight into the genetic sex of an individual. We can learn about whether someone had X chromosomes and Y chromosomes or only X chromosomes in their body. And generally, if individuals have only X chromosomes, they are typically identified as female. If you have an X and a Y chromosome, typically individuals identify as male. But the DNA can't tell us what the gender identity of that person was. It can only tell us what their genetic sex was and which chromosomes were present. So there's some information to gain there about sex, but some limitations in terms of what we can learn about gender. Similarly, I think in terms of ancestry and relatedness, the DNA can give us some direct windows into the biological relationships or genetic relationships among individuals or groups of people. In archaeological context, for example, we may often have questions about whether different groups of people who share material culture or share certain cultural objects may have been closely related. And in some cases, we may see different communities exhibiting some of the same cultural traits because they're closely related to one another, or it may simply be that they share those objects through trade interactions but are not closely related genetically. And the DNA can help give us some direct window into those biological relationships to see are the DNA sequences of people from these two communities very similar to one another, or are they quite different? We can gain similarly some insight into past migrations of people among different locations in the world, because if we see individuals or groups of people in different locations who exhibit similar DNA sequences, we can then infer that there may have been movement from one of those locations to another in the past. And that can then help us understand, for example, where people may have come from if we know that they moved in the recent or more distant past. There are some other ways by studying dental calculus, for example, we can gain some insight into what ancient peoples may have eaten, what their diets may have been like by looking for DNA from the plants or animals that they were consuming. And then we can also, in some cases, learn something about the lived experiences, what kind of stress, how much trauma an individual may have faced, by looking not specifically at the DNA, but at what are known as epigenetic markers, or these kind of other molecules that sit on the surface, on the backbone of the DNA, and the presence or absence of those marks on the DNA in different parts of our genome can sometimes give us insight into whether an individual may have experienced a lot of stress or trauma during their life or less so. Sam or Raquel, if you want to add to this at all in terms of what we can and can't learn.
I just wanted to bring this back to the original question that Sarah asked in terms of the relationship between ancient DNA and archaeology, because what the information that Deborah is talking about, we can use the molecular data, what we can actually learn from DNA in context with what archaeology is really good at learning about via materia culture and various other dimensions of what archaeologists study. And in context, the molecular data can then actually tell us a lot, right? And so it's only in conjunction with and in collaboration with archaeologists that I truly think ancient DNA is the most powerful, right? Because when you're just looking at the genetics, when you're just looking at the DNA, it doesn't make nearly as much sense as when you're collaborating directly with an archaeologist who can give you further insight into the context from which the remains were discovered. So for one example from my own dissertation, there are individuals that we're interested in learning both about their genetic ancestry, but the local community in Austin, Texas also has questions about where these individuals were originally from in just their lifetime. DNA can't tell us anything directly about where someone was born. You could maybe take a guess, but for example, you wouldn't be able to tell from DNA that somebody was born in Connecticut and then at some point in their life moved to Texas. There's no way for genetic information to tell us that, but isotopes can tell us something about where someone was born, where they spent the first couple of years of their lifetime. And so in conjunction, you can, one, understand where someone might have been born or the general area that they might have been born in using isotopic data. And then using the DNA data, you can gain insight into their genetic ancestry, which gives you a picture of where their ancestors came from. So whether it be Europe or Eurasia or Africa. And so in context, that gives us such a better picture of the individual themselves than if we were just looking at the genetics.
And this is
Yeah,
Raquel here. Oh, is that all right if I add one more point?
Sure.
I just really want to echo the results that the points that Deborah and Sam have raised, that DNA is really powerful in concert with these other forms of evidence, but that also we have to be really careful to not overinterpret the DNA evidence. Yes, DNA is an extremely powerful tool to be able to understand the ancestries of burials and deduce if anybody is related to each other, which other archaeological methods can't get at without direct documentary evidence. Unless we know what the headstone of this individual is and what their name is and what their names are, we can't get at those levels without DNA. But we also have to remember that DNA and how we interpret DNA is also a byproduct of the methods that are used to produce it. And this is where we get into conversation about, let's say, the genetic ancestry testing, 23andMe and Ancestry DNA, where you send your swab, your spit or your buccal swab, your cheek swab in, and then they give you a set of results that say you are 30% Irish or whatever the heck you are. And so we have to be really careful and cognizant as geneticists that when we're interpreting this genetic evidence, we talk about it broadly. We talk about it with understanding how are these results produced. So, for instance, like with genetic ancestry of archaeological burials, we will compare the DNA of that archaeological individual with reference populations from across the globe. and we have to be very careful. It says, well, according to, in comparison with these specific reference populations, this is the type of ancestry or the origins of this archaeological individual. So again, it's really being careful to interpret our ancient DNA evidence in line in concert with all these other forms of evidence, all these other forms of knowing about the past, but then also being really cognizant not to overinterpret our results as geneticists.
My understanding anyway, like the ancestry test, right? As their population grows, they can get a little more fine-tuned. Is that what we're going to see with ancient DNA? Will the population comparative groups that you guys are using get more well-defined, and you'll be able to maybe add a more specific element at some point in time?
I think so. Take, for instance, work with African diasporic populations and archaeological individuals. Africa is home to the most genetically diverse set of human populations in the world, and yet they are chronically undersampled. We have very limited understandings of the genetic diversity of populations in Africa and really across the world. Most ancient DNA sites have focused on Europe, and I think that is also a direct reflection of the contemporary reference genomes that are also mainly from Europe as well. So I know a lot of efforts are right now underway to ethically sample and work with communities in underrepresented places, so then in reflection, our work can also try to be better. And also, trying to move away from more racialized understandings of ancestry in these genetic ancestry tests, where somebody, and really going more by population, is really critical to having a more nuanced interpretation of these results. And I don't know if Deborah and Sam want to jump in and add more to this.
Okay, well, I think you explained it pretty well.
Yeah, thank you.
I think that, you raised the concept of ethics, and I think that this is a line of inquiry that raises a lot of potential ethical questions and ethical dilemmas. And we know, and I sort of raised this a little bit earlier, but I think that we've talked a lot on this show about the fraught relationship between anthropologists and different marginalized groups of people due to exploitative research practices and colonialism and the collection of human remains and artifacts in contexts that were oftentimes unbalanced in terms of, power differential and things like that. So DNA research must raise special challenges because it's a physical piece of a person, right, whether you're extracting, but also there's a lot wrapped up in genetics and identity in terms of real-world consequences. So can we talk a little bit just about, the ethics of DNA research?
I think that one is going to me. This is Raquel again, and I also invite Deborah and Sam to chime in after this as well. I think you really summarized it really well, that when we're working within science, we also work with a lot of inherited histories, right? So these are histories of colonialism and enslavement that are continuous processes that enact upon populations today. I mean, the structure of our society. So us as researchers, we have to be really cognizant about how we engage with science, right? How we engage with genetics, understanding that genetics has an inherited history of being a racialized science, has backgrounds just like, biological anthropology does, or biological anthropology is the field that all of us are in right now in this conversation, where, we've compared skeletal morphology in terms of racialized hierarchies, with European persons being at this top of this quote-unquote hierarchy, and so we have to be really aware that when we're talking with communities and talking about these results, that we're not re-racializing genetics, right? And this kind of harkens back a little bit to the point I made earlier about we're talking about a very tiny, tiny fraction of our total genomic diversity is actually being analyzed in these individuals. And populations, not races, share this continuum of genetic variation that spans and is related to these out of Africa migrations and expansions across the globe. So when we're thinking about these inherited histories and how do we translate these inherited histories to actually on the ground work with communities, really I think it's really critical to first listen to communities, hear about what their concerns are, what their questions are, what their needs are, and really just engage in this active form of listening. So you can then reflectively consider, well, how can I make this research attentive to these communities' needs? How can I make this research more sensitive to what their concerns might be? So, for instance, in Charleston, South Carolina, we did a lot of educational initiatives, a lot of public programming, a lot of school visits to really explain and break down what the process of doing ancient DNA was, to show people, to take away the black box that is science, to try to build trust through transparency. I think this aligns a lot with work that's done in public archaeology, where folks are really open to sharing about how we do this process so that folks understand and can build a relationship with us as researchers, but also with science as well. So I think, with listening, then also being accountable too. So once we listen to communities, which is an active and ongoing process, making sure that we come back to communities, report results, and really follow through on research promises. And this type of, I like to think about this as kind of a long-term relationship building between scientists and community members to really try to be aware of these histories, but also try to write new histories in a light that is founded upon these principles of trust.
One of the reasons why we love this program on ICRV Radio, and the one way that it's set apart from the 50 other programs on our air, is not only are the guests wicked smart, Deborah, Raquel, and Sam, not only are the hosts wicked smart, Sarah, Scott, and Glenda, but our listeners are so smart that they ask questions that I can't even read. So I'm going to bring Scott in to read a listener's question.
Yeah, it's a great question. It's from Tom. It says, I'm intrigued by, and it might actually kind of touch on a little bit of this ethical conundrum we were just talking about. I'm intrigued by the concept of ghost populations suggested in the ancient Native American DNA. It raises questions as to how far back the ancient DNA data provide insight into population ancestry. Also, what are the limitations or current constraints in analyses and interpretation? In a 2018 publication, SAA paper in a general overview book by David Reich titled Who We Are and How We Got Here, Ancient DNA and the New Science of the Human Past, it was noted that within 2 Amazon basin Native American populations, traces of genetic materials more closely aligned with Australasians than other population groups. The detected traces are only a small percentage of the genetic makeup of these populations, hence the ghost population moniker, possibly suggesting of ancient ancestry. The implications are that there are very early population with a common Australasian ancestry had moved into the Americas and then was replaced or absorbed by the later first Americans. Do you guys have any comment or anything you can kind of maybe throw some light on the work that you're doing on that question from Tom?
I can jump in here. I think this is, I guess maybe the first thing I'll just say is that I think it's important or maybe worth recognizing that some of the publications and work in this area can get into a lot of technical detail and very fine-scale inferences about the identities and boundaries and differentiation among peoples who lived in different parts of the world in the past. And a lot of this is interpretations that come from very complex and nuanced analyses, and some of these insights may or may not be a good representation of what really existed in the past. It's scientists' best guesses based off of the data they're analyzing. I think broadly what I will say is none of the three of us are working specifically on research related to the very earliest migrations and dispersals of people in the Americas, but what we do know from work that I've been involved with, from work that other researchers have been involved with, is that the history of communities in the Americas, like every other place in the world, is quite complicated and complex. And it hasn't been a simple story of one group of people spreading out across the Americas, but rather it seems that there has been multiple movements of communities into the Americas at different points in time. There has been movements of people back and forth between Alaska and Siberia, for example, at different points in time. And the series of movements as people spread throughout North America and Central America and South America has also been quite complicated. In some cases, we can see very sort of clear lines of relatedness between present-day communities and ancestral communities in the same region. In other cases, when we look at ancient DNA from ancestors who lived and died in certain places in the past, we may see genetic markers that are not so prevalent in any communities that have been studied today. And so in some cases, researchers have interpreted those signals as what they may call these ghost populations, or essentially a signal of some group of people that isn't as well known or well identified today, but may have lived in a place in the past. And I think it's certainly reasonable to expect that there have been groups of people that have lived in places that we may not have clear traces of anymore. But I think it's also important to recognize that there has been some real disparities given the history of genetic research in terms of which peoples have been well studied and which ones have not been as well studied. And so in some cases, when we see genetic signals that we haven't seen previously, that may be because it represents a community of people who are no longer present in the world, or it could be that it represents connections to descendants today that just haven't been as well identified because some communities have not been studied either because they don't wish to be studied or because that research simply hasn't happened yet.
Yeah, thank you. I think that's a great answer.
So I know we only have about 10 minutes left, but I'd love to hear a little bit about some of a little bit more about some of the research that you're all doing. Would you want to do just sort of a quick roundtable of what you're involved in terms of research projects and just to kind of give us a good sense of the variety of types of DNA work that the Yukon lab is involved in?
Yeah, I can go first. This is Raquel. So I completed my dissertation back in June from the University of Pennsylvania. And so my dissertation research looked specifically at two sites. One of them is in the colonial Chesapeake, and it's a 1680 site on the Delaware frontier. So if anybody has gone down to the Delaware beaches, this is kind of near Rehoboth Beach. So it's quite a nice spot to go check out. And it's really a story of family, and it's also a story of how folks were navigating enslavement at this time. There are 8 individuals of European ancestry and 3 individuals of African ancestry. And what our recent findings that have not yet been submitted, so we have to keep a little bit on the DL, we find that there's a little bit of a 3-generational relatedness between a grandmother, a mother, and a child between the European individuals, and evidence of a father-son relationship between the African individuals. So this becomes a really powerful way to both deep in the archaeology and the site interpretation of, but then also thinking more deeply about, well, what was these lives like for these people? How was the site occupied through time? So these are the types of things that DNA can help illuminate in terms of relatedness and ancestry. And I'll actually talk a little bit about a project that we're working with Sarah on to develop, which is a DNA pipe stem project from the Hollister site local in Connecticut. And amazingly, and I just am always so grateful to work with archaeologists that are cognizant of, potentially using future methods. In the plow zone fill of this Hollister site, there are a lot of tobacco pipe stems. So these are little clay, very common at archaeological sites, and these pipe stems are found in this fill. And so Sarah has put them in a refrigerator, which is DNA land is amazing because basically it means that we preserve and kind of keep stagnant all the microbial DNA and all the DNA that could be embedded in those clay tobacco pipe stems so they don't continue to degrade. And so what we're hoping to do with Sarah and in concert with her stakeholders is to try to develop a protocol on how we extract DNA from these pipe stems in a more robust way. There's one paper that came out in 2019 that they did this at a site in Maryland, and it was really successful to understand African origins of this person who smokes this pipe. And so our thought is that the utility of this is that, well, when we don't have burials at these sites, who is here? If we don't have good documents, maybe potentially clay tobacco pipe stems might help us think about occupation, turnover, or is the same person using the same tobacco pipe stem, or are they just being thrown away and being used by another person? So things in methods development are ongoing, but we're really excited to see if we can get it to work this summer.
Yeah, and I know Mark Packard is jumping up and down right now.
Yeah, it's a project he's been pushing for a long time, so we're really excited to finally be doing it.
I guess I can jump in and just talk a little bit about my dissertation research. Y'all are getting a fairly biased sample of ancient DNA research today because Raquel and I are actually, our research is very similar, and for most ancient DNA researchers, they're looking at individuals who were alive a little bit longer ago than the recent colonial past to about 100 years ago. But Raquel and I both work in contexts that do require us to take a more historical archaeological approach. And so my dissertation is looking at individuals from what we'll call, quote-unquote, forgotten burial sites. So these individuals were buried, and then for a variety of reasons, their graves were never marked, or they were never designated specifically as a cemetery. And so in these two separate contexts, again, one in the city of Austin, Texas, and then the other one in a suburb of Houston, Texas called Sugar Land, these individuals were, again, quote-unquote, discovered due to construction projects. And during these construction projects, between the two sites, 134 individuals were uncovered. And in some cases, we don't know a lot about who these people were. For the Sugar Land site, there is far more archival documentation of these individuals because they were associated with what's known as the convict leasing system, which was a system that kind of proliferated across the southern United States after emancipation, after the Civil War, where incarcerated individuals were used for labor in terms of building railroads and different buildings. And in the case of these individuals, they were actually, quote-unquote, leased out by the state to harvest sugarcane. And so we do have some archival documentation of those individuals simply because they were associated with the penal systems of Texas at the time. And then the individuals in Austin were actually found in a part of the first city cemetery of Austin that a chapel had actually been built on top of their bodies in 1914, and in 2016, the city of Austin did they were doing a renovation project on the chapel and discovered that there were 36 people that were buried under the chapel. And so, not knowing that they were there, community members in Austin are now, obviously, for very obvious reasons, interested in who these people were, but there's not as much archival documentation associated with them. And so between these two sites, one, a really big interest of both communities in Sugar Land and in Austin is to see if there's any direct relatedness between living descendants today and the individuals that have been uncovered between these two burial populations. And additionally, we're also interested in seeing what we can learn about the lived experience of these people. And so between collecting data from living individuals and as well as looking at what Deborah was mentioning a little bit earlier about epigenetic markers and trying to understand some of the molecular correlates of stress and trauma as it's related to things like penal labor and as it's related to a very specific time period in Texas's history when it was actually becoming a state. I'm interested in seeing what the correlates of lived experience are and how they're inscribed into people's bodies via their genome. So that's kind of quick and dirty what my dissertation is about.
Got 2 minutes left.
Thanks so much. I mean, I feel like as always we get to we get right into it, and then suddenly the show is almost over. So I know people are probably interested in learning more about this. Could you all maybe just provide, contact information or websites or anything like that where they can learn more about your research as we close out the show?
Yeah, the one option is to visit the Anthropology website, the Department of Anthropology website at the University of Connecticut, which is anthro.uconn.edu, and there's some information there. And then there are, I think, at least one other website that's kind of in development related to the Jamestown project, but I don't think it's public yet, but will be soon. And Sam and Raquel, do you want to add other sources?
Yeah, we have our personal websites. Mine, you can find me at raquelfleskes.com, and that has some more information about work down in Charleston, which we didn't really get to talk too much about. And then in the future, we'll link for that Jamestown website that Deborah was talking about. And this is going to be a website that focuses on how we do ancient DNA. So there's going to be future videos and pictures about the lab. So if you really want to learn more about how we do this process, stay tuned.
Sam, quickly.
I would also say stay tuned for website and more online content about my work. It's kind of all in process right now.
Thanks so much, guys.
Yeah, thank you so much.
As always, you can learn more about what OSA is up to at our Facebook page and at our website, osa.uconn.edu.
Yeah, and Friends of the Office of State Archaeology, look us up on Facebook or follow us online.
Thanks, everybody.